Cell adhesion molecules control interactions between leukocytes and endothelial cells during an inflammatory response. The recruitment of activated phagocytes to sites of infection is one of the most important functions of innate immunity. Recruitment occurs as part of the inflammatory response and is mediated by cell adhesion molecules that are induced on the surface of the local blood vessel endothelium. A significant barrier to understanding cell adhesion molecules is their nomenclature. Most cell adhesion molecules, especially those on leukocytes, which are relatively easy to analyze functionally, were named after the effects of specific monoclonal antibodies against them and were only later characterized by gene cloning. Their names, therefore, bear no relation to their structure. For instance, the leukocyte functional antigens LFA1, LFA2, and LFA3 are actually members of two different protein families. Three families of adhesion molecules are important for leukocyte recruitment. The selectins are membrane glycoproteins with a distal lectin-like domain that binds specific carbohydrate groups. Members of this family are induced on activated endothelium and initiate endothelial leukocyte interactions by binding to fucosylated oligosaccharide ligands on passing leukocytes. The next step in leukocyte recruitment depends on titer adhesion, which is due to intercellular adhesion molecules, ICAMs, on the endothelium binding to heterodimeric proteins of the integrin family on leukocytes. We have already encountered two of the leukocyte integrins that function as complement receptors, CR3 and CR4. The leukocyte integrins important for extravasation are LFA1 and MAC1, another name for CR3, and they bind to both ICAM1 and ICAM2. Strong adhesion between leukocytes and endothelial cells is promoted by the induction of ICAM1 on inflamed endothelium and the activation of a conformational change in LFA1 and MAC1 in response to chemokines. The importance of the leukocyte integrins in inflammatory cell recruitment is illustrated by the disease leukocyte adhesion deficiency, which stems from a defect in the beta-2 chain common to both LFA1 and MAC1. People with this disease suffer from recurrent bacterial infections and impaired healing of wounds. And that's cell adhesion molecules control interactions between leukocytes and endothelial cells during an inflammatory response. Have a great day.